All right, so for this video, we're going to talk about the parts of the cell and membrane junctions. It might be longer than others. So relax and let's talk about the cell and the important parts of the cell. All right, so the parts within the cell can be divided into either membrane-bound or non-membrane-bound parts. What that means is if they're a part of the cell like the mitochondria right here, if they essentially have another membrane specifically around them and have a separate internal environment compared to the cell, then they're membrane-bound. The endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and mitochondria are all examples. The first, the endoplasmic reticulum is outside of the nucleus. It's one continuous lumen, which means the lumen is like the opening in the middle. So it's all continuous with each other. There's no breaks. And extends from the outside of the nuclear membrane all the way to the edge of the cell. And if it has ribosomes on it, it's called rough ER, and if it's without, it's called smooth ER. And here is the nucleus, like I said, and the ER is outside to that and continues towards the plasma membrane. The rough ER is right next to the nucleus and has studded with ribosomes, and the smooth ER is outside of that, and it doesn't have ribosomes. These figures in your book that are talking about the parts, these little things, really gives you the gist of what the parts are for. So if you don't have time to read every detail again, maybe just take a look at these charts. Here is a little picture of where the rough ER looks like when the nucleus is intact and when part of the nucleus is cut out. Again, this is just cross sections of underneath the microscope. You can see the rough ER here, and the dots here are the ribosomes. The rough ER, the ribosomes themselves produce proteins. So the rough ER is involved in protein production. It's going to ship out proteins that it's making via transport vesicles, so little pods, little vesicles that send out the ER. They go from the ER, rough ER, to the Golgi apparatus. Make sure you know that they're connected that way. The rough ER helps produce other parts of the cell too, like the peroxisomes. And there again, here's the nucleus, here's the rough ER. The smooth ER doesn't have ribosomes. So the rough ER does have, the rough ER compared to the smooth, there's also a different purpose. So the smooth doesn't have ribosomes, so it's not involved in protein production. It's going to make fats, transport them, and store fats. It's also going to be involved in making metabolizing carbohydrates and detoxing drugs, poisons. There's a lot of smooth ER cells in your liver and then in your testes cells, because testosterone is a lipid, and so there's a lot in your testes. And here just shows where the smooth ER is in relation to the rough ER in the nucleus. The Golgi apparatus, it's composed of cisternae, that's what it's called, elongated sac-like material. And there's two different sides, depending, because it's polar. So there's going to be a cis face that's closest to the ER and a trans face further away. This shows it. Okay, so here's the whole thing, the whole Golgi apparatus. The cis face of the Golgi is closest to the rough ER, and the trans face is further away. So the Golgi apparatus is receiving proteins from vesicles that have come off the rough ER. They're going to take those proteins, we're going to modify them, package them, and sort them. The nickname of the Golgi, make sure you know, is the post office. And how it releases the proteins, again, is in vesicles, secretory vesicles that usually take that protein and it comes out of the cell. So vesicles become the plasma membrane and released outside the cell. This just shows protein or Golgi apparatus underneath the microscope, the cis, trans, and again, everything Golgi apparatus is highlighted in blue. Here's from your book that shows great images of the Golgi apparatus, different sides of it, what it looks like, what its purpose is. This one I really like down here because it's connecting the rough ER, the proteins that are being produced, sending it to the Golgi apparatus. The proteins in the vesicles being sent outside of the cell. The lysosome is a circular structure, sac-like structure. It contains very important digestive enzymes. When these enzymes are combined with certain parts of the cell or certain things brought into the cell, they digest them. These are like the garbage disposals of a cell. There we can see them where they're located, lysosomes. Peroxisomes are very similar to lysosomes. They're membrane-bound sacs. They also have enzymes within them, but there are different functions for those enzymes. These enzymes are going to participate in digestion, oxidation reactions, and making lipids. So the functions aren't to digest bad things, but to just in food digestion and making fats. Peroxisomes. Interesting to note is a disorder called Tay-Sachs disease. It's an inherited genetic disorder in which they don't have appropriate enzymes in their lysosomes, so they fail to digest lipids accordingly, and lipids accumulate in the nerve cells, causing the results of the disease, blindness, deafness, early death. All those things I just mentioned together, those are the membranous structures, and they're kind of all connected in transporting things within the cell. Mitochondria is also membrane bound, but it has a very unique and different role. There's what it looks like in a drawing, and then underneath the microscope. The outer layer and the inner layer. These little folds are called cristae in the middle, and the fluid in the middle is called the matrix. Mitochondria are involved in cellular respiration, so producing energy within the cell. So they're nicknamed the powerhouses of the cell. There's it shown in those images, the green. There's the cristae, and the outer, inner area of the cristae, and the matrix. The mitochondria are going to be more plentiful in cells that have high metabolic needs or need to make a lot of energy. Muscle. Ribosomes. So I mentioned those when we talked about the rough ER. They're attached to rough ER sometimes, or they can be free. They contain protein and a specific type of acid called ribonucleic acid. To make a ribosome, it's arranged with a small and a large subunit put together. You don't need to know the details of this. I'm just going to delete it. They're actually manufactured in the nucleolus of the nucleus. And they're made in the cytoplasm. So some can be bound to rough ER. And their job is to make proteins. So there's what they look like. There's free ribosomes. This is what they look like when you combine the large and the small subunit. And they make proteins. Cytoskeleton is similar to the purpose of our skeleton, our body. It gives support to the cell, help organize the parts. Cytoskeleton also has specific kinds that are involved in cell division and movement of materials within the cell. They're going to anchor the plasma membrane, help it stay supported. They include microfilaments, intermediate filaments, and microtubules. This just shows, and they're arranged right here from smallest to largest. So microfilaments are the smallest. An example, a very important example, is actin, which is involved in muscle contraction. They help separate cells during cytokinesis, or the division of cytoplasm. Here's the last part that I mentioned, participate in muscle contraction, because actin is an example. So again, many diverse functions. Microfilaments are the smallest. Intermediate, they're a little bit more rigid. Again, they are involved in structure. They're going to support cell-to-cell junctions. An example of where we might find them Keratin in the skin gives a hardening, thickening of the skin, hair, and nails, which is probably the most interesting one to note. There's intermediate. And then microtubules are the largest. They're hollow cylinders, kind of like a churro. And they're composed of a protein called tubulin, microtubules. They can be elongated or shortened, and again, they're involved in cell shape, moving things around. They actually form the cilia and flagella. They're going to move things around within the cell via vesicles. And they also separate chromosomes during cell division, pull them apart in spindle fibers. So there's what they look like. Really kind of cool pictures. Filaments, microtubules. So you can see they're spread within the cell. Here are the centrosomes, which are made up of filaments. And the size difference right there. Centrosome. We just saw them on this image right here. Centrosomes they're a pair per animal cell, so 2. They're oriented in cylinders. Their primary function is to organize microtubules in the cytoskeleton, and then they do participate in cell division. They move to the opposite sides of the cell to separate the chromosomes. So there's the pair of centrosomes. They kind of look like churros, I think, underneath the microscope. Here's their cylindrical orientation. This is a cross section or a transverse cut. This would be a longitudinal cut. Isn't that cool? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 groups of cylinders combined. Looks like there's 3 groups in each area, just like that. I think it's so cool that through microscopy we figured that out. What their arrangement looks like. Proteasomes. They are barrel-shaped proteins. They digest proteins that are probably inoperable for whatever reason. So they take care of damaged proteins or incorrectly folded proteins, proteins that you no longer need, and they are just can take out proteins in the body. Proteasomes, so they're complexes that digest proteins. Here's what they look like, kind of like barrel-shaped. And the proteins go in there and they essentially recycle the parts of a protein to build new proteins. The cell also has cellular extensions, so not within the cell but attached to the plasma membrane, cilia, flagella, and microvilli. Their cilia and flagella are involved in movement of some sort, and microvilli just going to increase the surface area for chemical reactions to happen in the plasma membrane. So there's what cilia looks like. There's actual pictures underneath the microscope, which is awesome. So there's cilia compared to the microvilli. Cilium, they look like cross-sectionally. So the difference between cilia and flagella that are both involved in movement. Different cells can have cilia lining the outside, the plasma membrane. An example, the cells that line your respiratory tract from your larynx, down your trachea into your bronchioles, during your lungs, are lined with cilia to move mucus and substances down towards the throat to bring it out of the body. And then flagella is a long whip-like extension. It's similar, but except it's longer, usually appear alone, like in the sperm, and its job is to propel the cell. In humans, the only example is the sperm, but in other cellular animals, organisms, there can be multiple flagella, like some bacteria. So that is the flagellum. What this is highlighting here in blue is different parts of the sperm, which you don't need to worry about right now. But the whole thing right here is this whip-like tail is the flagella. Microvilli, the nickname is called the brush border. They're just microscopic extensions of the actual plasma membrane. They're very, very, very small and very densely packed. They don't have any kind of power movement to them.